Uh, First of all, thank you so much, Prasanjit, for inviting me to do this talk. Very happy to be here and happy to see so many people over there for the talk. Um, I only have half an hour, so I'm going to kind of go at a fairly fast pace. But in case you do not understand anything or you have queries, please feel to put it in the chat and then we can take it on from there and I can explain further. So yeah, I'll dive straight away into the presentation. So I've been asked to basically give a brief introduction to citizen science, which I would also like to do, and then go on to give a demonstration of the portal. Uh, I can't see. I mean, I'm full screen, so I can't see you. But please feel free to stop me at any point if there's any uh, disturbance or you cannot hear me or anything. So my name is Thomas Vatakavan. I've been working with the India Biodiversity Portal, and we've been leading the development of the portal since 2008 at Strand Life Sciences, where I'm working. So, I mean, it would be difficult to start the presentation without asking what is biodiversity. And I'm sure all of you know biodiversity, which is the very reason why you are all here. And if you, we all know biodiversity is simply the variety of life, to put it simply. If we look at the definitions, they all refer to the variety of life all around us, so plants, animals, insects, everything. But if you try to go and understand it better, why are we studying biodiversity, and what exactly is biodiversity? Is it only the variety of life? Then you'll see that there are definitions which go into a slightly deeper understanding of biodiversity, and this includes the habitats around us, the different species, the different ecosystems, and the different interrelationships between organisms. I'm sure all of you would have studied this in your zoology or botany lessons, or generally in science. So basically, what we are referring to is the web of life. I'm sure all of you have heard the web of life in food chains, food webs, and all these things. But let's try and understand it a little bit more than that, because those are from textbook definitions of, what is the web of life. But basically, what we're trying to understand is what are the species out there in a particular habitat. What are the different types, and how are they classified according to our study of science? Are they producers, consumers, carnivores, herbivores? Are they parasites? Are they commensals, etc., etc.? How many of them serve as food for others? There's a lot of lot of things going on in the web of life, and we can ask so many questions if we take the entire species and try to understand it. But the basic thing is, what are the species around me? And that is dependent on sciences like taxonomy and nomenclature. What role do each species play in that web of life, or in generally in the whole ecosystem? This is basically related to ecology. How does each species interact with each other? There are things we know. We've classified them into positive, negative interactions. There are unknown interactions which we still don't know today, which we would like to try and understand better. What do they eat gives us a huge insight into how they interact. Where do they occur? Do they occur in specific niches? Do they occur generally all across the world, or all across India, or are they dependent on a particular temperature, or a season? When do they occur? What factors affects their lives? These are still things that we fully don't know yet, and these are things that we need to understand. And basically, what I'm trying to say is, in order to understand our web of life, or the web of life in our ecosystem all around us, we require data. I'm sure as a scientist or as a student, you will agree with this. Without data, we cannot begin to understand. We can observe, we can try and understand it better, but without having enough data, we cannot go in and study these creatures better to understand and make life better for them and for us in any way. So that brings us to biodiversity data. So what is biodiversity data? It's a huge spectrum of things, right? But basically, it distills down to, at its most basic unit, it distills down to taxa, space, and time, or what, where, and when. And this is what we call as primary biodiversity data. The basic unit of biodiversity data is primary biodiversity data, which says this particular organism or this particular individual of a particular species was found at a particular place and a particular time. And that's what we refer to as observation data or occurrence data. Then there's also secondary biodiversity data, which means that if you take all the primary biodiversity data, you can begin to derive other information from this primary biodiversity data. The things of distribution maps. You can publish reports saying what is the abundance of species, or what are the rare species found in a particular place, or interactions, infographics, and all these things. These are basically secondary biodiversity data. So from primary biodiversity data, you begin to aggregate, put together, analyze, and you arrive at secondary biodiversity data. Now, typically, you as a contributor of biodiversity data will have something like this, a photograph of a pretty photograph, an interesting photograph. You can see here, it's a spider eating a fly. And this is a good photograph. I'm sure a lot of you have seen, if you're remotely interested in insects or life itself, or even just for photography's sake, you might have taken a photo like this. And this photo is a nice photo by itself. But how do we go from turning this photo into data, right? So when you take that photograph, you know for sure you took it at this particular date, and you know that you took it at this particular location. So here, 29th March is the date, and 25.1849 and 75.8250 are the latitude, longitude coordinates, which place that the recording of that particular image at a particular location. So suddenly, from just a pretty image, you have data. You have some kind of data saying there's a place and a time. Now, what happens if you are able to identify this organism, or both the organisms, if even better? Now, if you are able to put a name to the spider, then suddenly it becomes more interesting. It becomes a point data or primary biodiversity data. It becomes something that is of interest to not just you as a photographer, not just people who look at it for the beauty of the photograph, but as a scientifically important occurrence point for that particular species. Now, what happens if 20 of you record the same organism from 20 different locations, or over different times or different spatial locations? And if you put this all together, suddenly it's of interest to scientists, because what's happening here is, from a pretty photograph, you're beginning to aggregate it and put it into what is known as distribution data for that species, right? Or aggregated data. And this is what we're trying to do on the India Biodiversity Portal and other portals. Try and get people to contribute data so that we can aggregate this information and study this organism better, understand it better. So yeah, I've shown you this photograph. I've talked about this much, but I just want you to think. I'll come to this point. But is there any other information that we can extract from the photo above? Think over it. I'll come back to it. So of course, we have to get into what is citizen science. So I'll quickly just say, basically, citizen science is public participation in science. So any kind of participation in the scientific research is basically citizen science. Whether you are just contributing an image with date and time, whether you are identifying that species, whether you are generating, analyzing data in combination with a lot of others, all these things are citizen science. So basically, what we are doing on IBP is collecting observations through photos, videos, audio, reports, etc. And we are getting you as a citizen to contribute towards this effort. So basically, I mean, it's very intricate. If you try to break it down, it's also known as crowdsourcing. It's called as PPSR, or public participation, scientific research, etc., etc. I won't go into that, but basically, IBP does the citizen science component by crowdsourcing observations through media and identifying the species and getting you to contribute the location and the date of the observation. And this we are using for further study, further generation of information from this data. So this is basically an observation. You can see somebody has contributed a snake. It has been identified as the common rat snake, and you can see it's from Tamil Nadu, India. We've recorded the date of observation, date of creation, etc. So somebody has contributed this, but basically from this we can then start to work out: can we use this image for something else? Can we use this image to create a page with information on that species, which we call as species pages, which is there on the India Biodiversity Portal? We can extract names and start to aggregate what this animal is called in both in scientific context as well as in different regions, in different languages. What do people refer to this species? Then if you have enough information, we can start to build distribution maps of this particular snake, which we have done on the India Biodiversity Portal. And this gives us enough information on, how common is this species across India? Where is it found? Where are you more likely to cite it? Where is more reports of it coming from? And that's just the basic level. Is it people citing this because of disturbances? Is it naturally found there? All that you'll have to analyze and see in combination with other types of data. And also from the date of submission or date of observation, we can then work out things like temporal distribution. What is the seasonality of this species? Is it seen across the year? Is it seen, just in particular seasons? Is it a migratory species? How rare is this in some seasons, and so on? So we can start. These are all secondary biodiversity data or extracted or derived data that we begin to put together from observations that you have submitted. So take a look at this. Again, another nice picture. I would have liked this to be an interactive session where I'd like you to answer some questions, but obviously I don't have time, so I'll rush through. So this is a nice photograph, and if I ask you what is this photograph, I think 80 to 90% of you will say it's this and this butterfly. But there's more information in this. It is once we begin to understand it, we know it's feeding. It's a male of the species. It belongs to a certain family, a certain class. It's an insect. It's an arthropod. It's a South Indian species. It's a species in trade. It's got certain characteristics in terms of color, the role it fulfills, and so on. But there's also one species below it, which is the lantana, the flower that you see, the plant, which typically goes ignored because people are focused on more visible, more attractive species, and they tend to not think about more things like what is the interaction happening here. And again, if you look at lantana, there you get a lot of information. If you try to understand it, it's an invasive species. It's an indicator of disturbed habitats. It's a food plant for several species. It's a fire-tolerant plant, etc. It's also cultivated ornamentally, and so on. And those are just basic bits we can extract once we know the species. But then there's also the interaction. So what is this butterfly doing on this particular plant? Is it feeding? Is it a food plant? Is there a relationship specific to these two? And things like that. There's so much information contained just within a photograph, right? And we can begin to, once we start to identify it and know where it's present, when it's present, we can put all these together and try to understand these relationships better. What I'm saying is that we can try to understand the web of life better through your observations that you submit. Yeah, so basically there's a lot of information there. And this we're trying to do on the India Biodiversity Portal, what we call as observation plates. We still only have a first cut of this, where when you are submitting an observation, we ask you to identify whether it's male, female, whether it's an adult, whether it's a pupa, larva, or if it's a flower. Is it flowering? Is it if it's a plant? Is it flowering? Is it fruiting? So that we can extract the phenology information or the seasonal information from it. What kind of activity was it doing when you observed it? What kind of microhabitat did you observe it, or what kind of habitat did you see it in, so that we can work out is a particular species associated with a particular habitat, and so on. And whether you're reporting wild or captive individual and things like that, so that we can weed out these things for later when we are analyzing the data. So basically what I'm trying to say is it's not just all about observations. It's not all just about occurrence data. There's so much more that we need to understand about biodiversity and the intricate web of life that goes on around a particular species. And that's what we're trying to do through the India Biodiversity Portal, because we have these different modules. We have different modules that collect different information. We're trying to put it all together, link it all together, and create more information on these, different species and different, types of information that come together. And that's how we've been building the India Biodiversity Portal, and all these intricate connections between these different modules, different types of data. So it's a very varied portal, very huge in its aim, which is also why it will probably seem a little bit complicated when you look at it the first time. But we're working on that. We're trying to simplify it and make it better for you to access. So, yeah. Sorry, was there a question? Okay. Okay. Yeah, so I'll skip this and I'll go on to a live demo of the portal because I have just 15 minutes left and I'll rush through. So basically, this is the India Biodiversity Portal. You can access it at indiabiodiversity.org, and you can, this is the homepage which shows you information on the different modules contained here. I won't go into too much detail because it's kind of self-explanatory, but if you have questions, I'm happy to answer them. Basically, we also have something here called groups, and these are the different groups or different micro portals within the India Biodiversity Portal, catering to different functional or different themes of interest. Among which, if you search here, Eco Club. So you can see we have created a group here for Eco Club SS Mahavidyalaya, which if you come onto the portal, you can go to Eco Club SS Mahavidyalaya and click on join the group, and you will be added to the group, and now you can start to contribute to the group. You can see here there are 42 observations. All I think all of them have been submitted by Prosenjit. You can browse this, interact upon it. You can help identify the observation. You can see 3 to 4 people have agreed on, some Prosenjit has identified it. 4 people have agreed on the identification. There are other things. I mean, you can click on identify. If you think it's a different species, you can suggest a different name. If you think it's the same thing, you can agree on that species by clicking the agree button. You can, if you have other groups on the portal, such as there's a butterfly, there's Butterfly India, there's Big Butterfly Month going on. You can submit observations to that as well. The same observation you can post it to multiple groups and so on. There's also this thing where you can click on the traits, and traits will give you any traits that are specific to that particular observation. I'll move on. Basically, I want to talk about how to upload observations, and so there are 3 main ways which I will quickly cover. You can do it by uploading a single observation, or you can, if you have lots of images, coming back from a field trip and you have lots of images, you can process that as a batch. Or you can, if you don't have images, you just have a checklist of species in an Excel file. You can upload that as a data table. So first of all, let's create a single observation. How do you do that? You can see here, the first thing to do is browse and select your pictures which you want to upload. And so here's a picture. Let me select these both of 2 dragonflies. Or a single dragonfly, let's say it's all from the same observation. You can select it and upload it. You can, of course, put in captions, and if it's not, you are not the contributor, if you are uploading on behalf of somebody, you can put in that information in the contributor. You can then identify it. So this is a paddy skimmer. You can, if you know the common name, you can type that in. You'll get a drop down. You can select that. It will automatically put in the scientific name and mark the species group. Dragonflies are arthropods, so we mark it as an arthropod. Next is, where have you seen this? So let's say you saw this in Kannur, Kerala. Select Kannur from here. Basically, what this is doing, you're typing in the place name, but behind the scenes, behind the scenes, if you click on advanced edit, you'll see that what we've done is search for that place name on Google and extracted its latitude and longitude and plotted that particular point on the map. And if you want to be more specific and say, I saw it actually at Kannur police station, you can move it there, and you can use that as a title, a specific title if you want. Use as title, it will replace this, or you can type in Kannur police station and leave it at that. But yeah, so you've added the location. You have a field here for location accuracy. You can mark it as accurate, approximate, or if it's something that you've forgotten, you don't know if it's a broader thing, you can mark it as a local or a regional observation and move on. It will automatically extract the location as well as date if it's already there in the photo that you've taken, but not all cameras support that. So, I mean, here you can see it, date is automatically extracted, and date accuracy you can again change, and so on. You can do other things. You can write notes about the observation. You can add tags. You can say that this is a human observation, which is the default if you're taking photos of it. But if you're uploading, living specimens or literature or machine observations or fossil specimens, you can appropriately mark that as well. Over here, you have traits, which I mentioned earlier. You can mark whether it's a male or female. In this case, it's a male. You can say that it's an adult. And again, here you have separate sections for complete and incomplete metamorphosis. This is a dragonfly. It goes through nymph stages and becomes an adult. So you can say this is the incomplete metamorphosis. You can say where you saw it. I saw it in a pond side, but it was also in an agricultural location. You can mark multiple. You can say it's very common in the area that I saw. You can mark that, and it's also a wild species. And here you have the option to add it to multiple groups that you may be a part of. You don't have to do that, but you can do that. And you can submit the observation. And if you submit the observation, it will create the observation on IBP. Yeah, it should get added. I don't know why it's slow. But anyway, I'll move on. I won't wait for that. The other way of doing it is uploading batch observations. Okay, uploading a batch observation. So here, the intention is, if you have a batch of images and you want to process that, you can select all the images. So browse, let's see, observation images, and select all the images that you want to upload, and click open. And it will briefly do some behind the scenes processing. What it's trying to work out now is how many of these observations belong to a single observation, how many of these belong to different observations. So you can see these 2 photos are a single observation, these 3 photos are a single observation, these are a different observation. It may or may not get it right. You can reorganize if it gets wrong. Simply drag and drop into another template, or you can even merge it back into a single template across. And you can say that there's an option here to detect and predict scientific name and species group. Again, these are machine learning. It will not always get it right. It's more difficult for some taxa than the others. You can say yes and continue. And now it will put it automatically into templates. So here, 3 observations with templates. If you can identify this, you can type it in. So this is common picturing, and this is the green marshot. So you can see it's automatically predicted what the species group is. It extracted the date and so on. Here again, the location you have to add. You can do that in 2 ways. You can individually add the location like we did earlier for single observation, or you can say select all, fill selected, and you can say use the last location that I've added. So this is what you added last time, and you can say save, and it automatically add the location to all of the images at a single go. You can also individually, if you want to add, you can say this is male or female. So go to traits, and you can say male, agricultural, pond side, and so on. I'm not going to do that, but That's how you add it. So any of these fields, or any of these fields, you can select all and add it at one go by filling it all. You can also merge. You can split observations, etc., etc. So if you want to split this, you can just select that and say split. It'll split it into its composite images, or you can merge it back by this. So all those actions you can do in bulk, and when you click upload, it will get uploaded, and it will be submitted as observations. So if you go here to observations, this is the observation list page. Here you are able to see all the observations that people have uploaded. If you want to see just your observations, you can click on your profile page and click on my observations, and it will show you only your observations. The other option is use the filters here. You will be able to filter by your own name, or you can filter by a particular species, a particular location, and so on. So you can do all those things. If you want a predefined location, you can choose by state, district, or tehsil, or you can draw over here and say I only want observations from this particular region, and it will give you that. Yeah, but moving on. So that's basically multiple observations. I have to tell you right at this point. You can, once you finish your query and you get your results, you can then click on download, and you can download these as a CSV file for further analysis. There are some, default fields that are provided. There are some fields that you can select to be extra columns in your data, and you can download that for further analysis. Finally, just moving on, what if you have a data table? What if you have a table like this? You've gone out and done a field study. You have a list of species with different dates. You have marked male or female. You've also added different latitude, longitudes for each row. You may or may not want to do that, but I want to upload this. So then you go to contribute, click on upload a data table, and select a file, checklist, and it will give you a preview of the file, the same file that you have marked up here. Now the important thing here is you need to mark which column is what. So your specific Excel sheet column, whatever you have called it, you just want to mark it to the database column. So this is a species column, and here are the options for the database column. So this is actually a scientific name. Mark it as scientific name. This is a date. This is a from date, because you can add a to date also. This is sex, and you can see if you go down here under traits, sex is there. This is place name, and the latitude, and this is longitude. And this is a column with images. I mean, typically you don't upload images for data tables, but we do have that option. If you want to associate images with each row, then you will have to have a column with the file names, the exact file names, including the file extension .jpg or .jpeg or whatever it is, and put that into a column. And you can call this, you can map it to file name column. And here again, since you're giving a set of data, we would like to know, or anyone who looks at it would like to know, what is this data about? You can give it a name, whatever you feel like, a survey from XYZ location on this day as a part of this project or whatever. You can write some description on it. You can then define between what dates have you taken it. Is it between 22nd and 28th? You can give a date range. You can mark it as accurate or approximate. And again, same, it's all data about arthropods, so I mark it as arthropods. Here again, a slightly different workflow for adding locations. You can actually add multiple locations, but typically you don't use that. So if you say this is all from Kannur, you Hello. Sorry, my laptop just crashed. Um, yeah, so just I'm on my mobile phone now. I'll just finish up in a couple of minutes. So what I can do from the mobile while I restart the laptop. Um, yeah, basically that's how. And once you submit it, you will be taken to the observation to the data table show page, from which you can each row of your data table becomes an observation. So you can further interact on that observation. You can re-identify it and so on. Or you can agree on names. You can change the ID. You can, all those things. Or you can get others to help you identify that observation like a normal observation. So that's basically a data table upload workflow. I don't know whether I can share my screen on the mobile. Yeah. Okay. Sure. So final thing, what Prasenjit required was basically a demo of the mobile workflow. Again, can you see the screen? Yeah. So basically, there is an app. If you go to the India Biodiversity Portal, there is an app available for Android devices. And if you go to the Play Store, or if you go to IBP, yeah, somebody's mic is, yeah, thanks. So yeah, so if I go to IBP, yeah, if I go to the website, you can see here there is a link to the Play Store. Okay, so basically, if you go to the website, there's a link to the Play Store, and you will be able to install the app from the home page of the portal. And once you do that, you can exactly in the same way that I mentioned, you can go about contributing data. It's actually what we call as a progressive web app or a PWA, in which uh, the workflow is exactly the same as the website, and everything is pretty much the same. So you can follow the same workflow that I mentioned earlier to also use the mobile app. The website will also load without the app if you go directly on the browser, and it will fit to the specifications of the phone so that you can use it as a unified workflow across both mobiles and laptops, etc. Finally, what I left out was the register workflow. If you click on the menu here, you can click on login, and on in the bottom of the login, if you already have a username and password, you can sign in using those credentials. If not, you can click on register. You just have to put in your name, select your gender, uh, put in an email number or a password or both. Here, we specifically recommend that you use an email because with mobile numbers, you don't get notifications of any activity, whereas with emails, you will get notifications on any activity you have performed or other people have performed on your observation. So use a email if possible, and put in a password, confirm your password. You can select what your profession and institution is, and finally, just put in a location exactly like you did in the observation upload workflow. I can say you're from Bangalore and select that, and select the captcha, and you're done. Once you click register, you will get an OTP, and the next screen will ask you to submit that. Submit the OTP, and you're done. You're registered. You can then go to your profile and edit it in order to make changes, upload a photograph, and so on. Yeah, I think I've covered everything. I'm just getting back on my laptop. So if you have any questions now, or if there's time, I can take questions. But back to you, Prasenjit.
Thank you, Prasanjit. Are you able to hear me well? Okay, perfect. Thank you so much. So I will just go ahead with my presentation quickly, and then we can discuss more. So, as Thomas just demonstrated, India Biodiversity Portal is a very useful website to mm, upload observational data using photographs or sound. Similar to that, there has been another initiative internationally called iNaturalist. So, once you check both the sides, there are a lot of parallels. So, iNaturalist also is like a online social network of people who are sharing biodiversity information with each other and trying to learn from each other. And it's basically some kind of crowdsourced species identification system and organism occurrence recording tool. So whatever you see, you post on iNaturalist, similar to India Biodiversity Portal. Uh, Create and maintain your own record of species, learn from each other, species identification, and in the process also contribute to science. So a little bit about uh, iNaturalist also has a website as well as app on cell phone. Uh, you can use whatever you want to use depending on what device you use to take pictures. If you use camera more, you can use website. If you are using cell phone to take pictures, you can use app immediately to upload it. So the reason why I use iNaturalist is it helps me to contribute to the global database and to the community science. Uh, I get help with identifications. It helps me build a personal life list, learn more about those species because uh, it has a particular mechanism of looking at the species pages. Uh, I can help others identify what they have observed wherever I know a little bit about butterflies and some other insects, so I can help them identify that. And I personally also use this data for my own research. So, have you, if you have not used iNaturalist, it's basically very similar to India Biodiversity Portal if you have used it. So, how it works is you record your observation and then you share it with your fellow naturalists, and then there is a discussion happening on iNaturalist website about what you have seen. So people uh, either confirming your identification, providing you identification, or generally asking more questions or giving you additional information about what you have seen. So very similar to any other social media like Facebook or Twitter. There are discussions happening. Only difference here is when you are talking of identifying species, it always is linked to the uh, database, what we call as the taxonomic backbone. So you are talking in terms of scientific entities rather than just exchanging comments in case of Facebook or anything else. So that is the advantage of using iNaturalist or India Biodiversity Portal rather than Facebook or WhatsApp and things like that. So basically, you can use iNaturalist to explore nature, to document nature, to publish or share your data. So there are different ways in which people use iNaturalist in. So here are some ways that are listed here. So you can use it to keep track of what you are encountering. You can use it to crowdsource identification. So you post something and you may have a very rough idea saying it's a dragonfly or a moth, but other experts can give you more precise identification. You can generally learn about nature. You can just browse iNaturalist, look at different records, look at different species pages, and learn. And in the process, you become a citizen scientist. You help in creating some useful data that other researchers can use. And also, you can run a BioBliss using iNaturalist. So BioBliss is nothing but trying to get a snapshot of biodiversity of a region. And if a group of people say visit a particular park and they decide to do a BioBliss, they basically um, collect all the different biodiversity records, take pictures and post it on iNaturalist or India Biodiversity Portal, and it it kind of creates a snapshot of what was there or what was recorded. So these are the different ways that iNaturalist is used for. So basically, at the end of the day, what is observation? An observation is like it's a record of an encounter of an individual organism that at a particular time and a location. So the important points here are uh, time, location, and what organism. And all these three combined together with a particular user, it creates observation. So this is a very basic unit of biodiversity occurrence data in some sense. So it is very important to know what exactly you are doing when you are uh, posting your observations on any of these citizen science biodiversity portals. So uh, to understand what exactly is a record, is here is a sample record where it has been identified as this particular banded brittle star. And so in this record, you can see there are a couple of pictures of this brittle star. You can see who has posted it. You can see when it was observed and when it was submitted. Sometimes there could be a difference between those two. I can always submit some of my really old photographs if I remember the details, like when I have taken it, which is typically in the file, but where I have taken it. And then there is the scientific information of what taxonomy is and a whole chain of identification and uh, comments. So this whole thing basically constitutes a biodiversity occurrence record. So as I was mentioning, there is community interaction on what exactly that photograph is showing. There could be people asking questions or informing what exactly is happening. So uh, you can see here there is a comment saying, looking at your picture, you cannot identify since it's a juvenile what species it could be, because I was trying to jump to a species. So then they said, no, it's very difficult to tell anything beyond family. So these are the kind of interactions and learning that happens basically uh, through the community identification. Okay, so the after there are a few identifications provided, iNaturalist has this mechanism of determining research grade. So the formula they use is there need to be obviously more than two identifications, and if there is anyone who is mm, challenging the identification for research grade, two-thirds of the identification should agree to a particular ID. So um, what happens basically once the record is research grade is it is ready for use for further downstream use. So iNaturalist basically then periodically sends these records to GBIF or Global Biodiversity Information Facility, about which we will discuss in a little bit. And also, depending on what kind of data it is, it also can send it to Global Biotic Interactions database. So this particular record of striped tiger was a butterfly feeding on a flower, and because the name of the flower and the relationship was specified, this particular record was also sent to Global Biotic Interaction database. So this is uh, this is the downstream use of the research grade records. Basically, iNaturalist indicates that this record uh, contains the basic information of date and place, and it contains identification. And now, any researcher who want to use it can potentially use it. So that is what is research grade in short. So when you are posting records on these portals, you need to remember certain things. So you basically need to avoid what we call as avoid 3 Ps. You should not uh, post pictures of people, uh, pets, and potted plants. People, for obvious reason, for privacy, and you don't have to record human beings. We all know where human beings are. Pets, because pets are not wild animals, but they are domesticated, and wild animal information is more information than other things. And then same for the potted plants. You can plant a potted plant anywhere in the world. So if you record potted plants, then the distribution will not be shown correctly. So it's not in some sense natural there, natural or wild. So that's the reason you should avoid these 3 Ps when you are posting. And as I was mentioning, what happens next is the metadata is shared with GB for Global Biodiversity Information Facility. Biotic associations are shared with Globi, and all the images that are sent, they are posted. They have potential of being used in the computer vision model, which helps you in identification of the records. So I uh, because Thomas has really given a very good demonstration of how to post records, I don't think I need to do the same, because most of the options are extremely similar. So I will not spend time displaying again the same sequences, but it's basically same basics that you need to input, enter, add a few pictures, make sure the date and the location uh, is geotagged properly. So I will now quickly move to second part of the presentation, where I'll talk a little bit about GB for the Global Biodiversity Information Facility. So both India Biodiversity Portal and iNaturalist share their research grade data with GB for the Global Biodiversity Information Facility. GBIF is nothing but international network of um, uh, different countries and different international organizations coming together to provide data infrastructure. So what we mean by data infrastructure is all this data that is shared on GBIF is made available in open access for any further research. GBIF also takes care of providing clear guidelines of how the data should be used. GBIF also provides guidelines and recommended citations. So any organization that is taking these efforts and publishing this data needs to be cited if the data is used in any of the downstream research. So that is the main goal of GBIF. So it provides this data to anyone, anywhere in open access so that um, people can use it. And in terms of scope, it is global and it is all kinds of biodiversity. So that is the scope for GBIF. So why should we publish the data on GB? So there are several reasons, and some of them might be applicable for you, some of them might not be. So here I am not only talking about sharing the data through individual records through portals like India Biodiversity Portal or iNaturalist, but I am talking about in general contributing data sets that those could be your own personal research data sets or your project data sets, where every time you may not have recorded photographs, but still, if you are working in the region and if you are a researcher, you know what biodiversity elements you're talking about, so you can make them into data sets and publish. So why you should do that is again very similar reasons like iNaturalist and India Biodiversity Portal. You're contributing to the global knowledge about biodiversity. The more the knowledge available, the more analysis and interpretation we can do. The data is from GBIF is available to a broader audience, both geographically as well as across the disciplines, because GBIF is very much well known and is being used for research over last several years. More and more is being published using GBIF data. So whatever data you publish, rather than just you using it, there will be a increased usage of your data. There are more and more opportunities to collaborate. If someone knows that you have certain data from certain region, they might approach you for newer opportunities for collaboration. Through GBIF, you can also track citation and usage of your data, so you can in some sense demonstrate the impact of your data. Suppose you are in a college or a university, you can show that okay, as part of your academic research, whatever data you have collected, these many people are using it from these many countries and things like that. So, and then what happens is once you publish data, in case people find any problem, uh, they typically provide you feedback. So your data also gets curated better, and you receive feedback, and you can improve the quality of your data. And in this process, you also improve the visibility of your institution. It could be your school, your college, your NGO. So these are different benefits of publishing data sets on GBIF. So what kind of data sources can be published on GBIF? Obviously, as we have been talking about, the geotag photographs by amateurs can be published through IBP and India Biodiversity Portal. But you can also publish your museum specimen if you have museum specimen collected for any project. Those can be digitized and published. You can, as I was mentioning, you can publish the data that is collected for any of your research studies. It could be a regular sampling of a plot for botany or regular bird or butterfly observations. If you are maintaining those records, you can definitely publish it. And if you are also involved with any of the new environmental DNA sampling, even that data can be published on GBIF as a data set. So these are the kind of data sets that you can publish. And now I will just quickly uh, jump to a demonstration. So I don't think I need to give a demonstration for iNaturalist. So I will only give demonstration of GBIF. Let me try and share my window. Okay, so this is the GBIF or Global Biodiversity Information Facility website. When you open, you are presented with this screen. It is basically telling you how many occurrences GBIF is currently serving. So currently across the world, it is serving about 2.5 billion records from 90,000 different data sets and 2,000 different publishing institutes. And this data has already been used in 9,300 research papers so far. So that's the amount of use that you see for GBIF data. So in these 90,000 data sets, we have the India Biodiversity Portal as one data set and iNaturalist as another data set, just to give you an idea what exactly are those. So I would highly recommend you to visit the GBIF website and go through it. So it basically tells you what is GBIF. It tells you what GBIF is up to these days. Some recent funding opportunities, some recent publications that are coming up. Some of the workshops are like one of the new things that they are trying out is they are defining standards for uh, camera trap data set. They recently did it for eDNA data set. So things like that uh, you can kind of learn and keep yourself updated. It also displays certain data use cases. So data can be used for health related things, for threat um, calculation, computations. You can do it for climate change. And these numbers here are the number of research articles using that. So there are 1,300 research papers published talking about invasive and alien species using GBIF data. So, is there a question or there is a raised hand by mistake? Okay, I think it is by mistake. We can take the questions later. Uh, so, uh, yeah, basically you can go through the website and see what they're doing. So these are some of their funded projects and what are the different activities they are doing. Then here are some of the projects that they are kind of highlighting on the GBIF website. So interestingly, uh, Prasanjit was talking about the groups like Butterfly India and Dragonfly India, Dragonfly South Asia. So one of the projects currently featured is actually one of our own group, that is the uh, Spider India group, and this particular data set is talking about the efforts that India Biodiversity Portal and Spider India group put together to publish a data set on GBIF. So you should take a look at it. There is this spider uh, data set that is published, and it has already been cited by in different research papers or projects. So this is how you can track once you um, uh, publish your data set. This is how you are able to track the usage. So now we can see that the spider data set which was published last year, these are the 18 research projects or articles who have used that data. So these are all the literature data sets. And then going back to the main website, there is also a very good resources section where you can visit. And this is one of my favorite maps showing how much data is coming from different parts of the world. So here, in some sense, it is a paradigm if you see the tropical region are is where we expect the most biodiversity, but unfortunately that does not have that much data published. More data is published in Europe and North America. That's because of the resources they have, and that is because of the active people uh, that are documenting there. But that picture is now changing because of projects like eBird and India Biodiversity Portal. Now you see even from India, more and more data is available. So to if I have a few more minutes, I will quickly show few more features um, of this website. So you have different different ways basically to search this particular data. Say suppose I'm interested in looking at data from India. So I can search for country India, and then it is telling me that there are 38 million records from India. I can look at those records on a map. I can look at those in some kind of density plots. I can look at the gallery of the images from that data. So when you're posting your records on India Biodiversity Portal and iNaturalist, your photographs also would show up here. I can also get an idea on the taxonomic coverage. So if you look at it, there is the whole data set is heavily biased towards animals versus plants, and if you look carefully, even within animals, you will see that most of the data is about birds, and that's because of the uh, eBird platform, which is supplying the data. So this is how you can basically explore the data. If you look at temporally, you see up to mid 2000s there is not much data, and then there is sudden spike in the data, and this is because uh, this is when the citizen science portals become really active. So these are, and you see about 99% of the records are human observations. So there are not many collections which have mobilized data. So these are some of the things that you can explore here to understand what kind of data is here. So as you can see from the data sources, out of what was the number, 38 million, 37 million are from eBird. There are about 220,000 from iNaturalist, and there are about 17,000 from India Biodiversity Portal. So this is how you can explore uh, any data. I think the opportunities are unlimited, and we can actually exclusively have a workshop of exploring GView data and how to use it. But in the interest of time, I think, stop here. I already spoken for about 30 minutes, and we can try and address some questions. Yes, so Two aspects. One is, even if you are not going to make a career in biodiversity or biological sciences, if you love nature, it's your responsibility to protect it. And as I was talking about, GBIF data is more and more being used for conservation or climate change related studies. So it is important that, I mean, it's 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 a whole ocean of information, and it's your responsibility, if you love nature, to add a few drops in terms of a few occurrence records. And even if it's a very commonly seen thing like an ant or a fly in your home, that is also an important data point, because sometimes you never know that people have not recorded that in your region. Or even seasonality is important. With climate change, things are moving. So that is one aspect. And the other aspect, as you were specifically asking, as I quickly showed you, your data set, if you publish, you can track the usage of it. So you can say that, okay, my, like I was showing the Spider India database, which we published last year in collaboration with India Biodiversity Portal. It is already cited in 18 or 19, whatever different research publications. So that is the impact that we have done already. And imagine how much impact we will have if we publish all our group data sets in the same fashion. Obviously, people are using and citing that data. That means they are finding it useful. And then there are mechanisms um, of tracking or checking where exactly and how it is being used. So if to kind of give a quick example, I'll share my screen once again quickly, and I'll show you something. So what it is showing currently is, you might be aware of this ORC ID, which is a researcher ID. So what I did was, I just typed in my researcher ID into one of these fields called recorded by ID. So which means that these are all the records that I have contributed to GB. So I have contributed 6,700 records to GB, and then I can look at my own gallery on GB. So when I click here, all these are the images that I have taken and recorded. I can look at the map. So you can see that most of my data is coming either from India or from United States. You can also guess in United States where I live because of high number of records. So this is the place where I have lived for several years. Currently, I am living here. So this is the place where more and more records are coming. But that I can also see that I have what other places I have visited. I have visited Costa Rica, I have visited Benin, I have visited Madagascar, I have visited Israel. And you can actually, yeah, so you can track your own life, but also you know that this much is my contribution to the, out of the 2.5 billion records of GBIF, I have contributed to so many records. And as I was saying, it will also track subsequently through different data sets how many people have used it for research. And similarly, this is not only what I have recorded, but I can also see what I have identified. So now what you see is, these are the places I have identified the record. So in a herbarium or in a collection, there is a determinant, the first person to identify something. So these are the number of records. So here you see they are from more wider geographies, because even if I have not visited, if I am familiar with that species, I can always identify that because I have that knowledge. So here, if you compare the map to this, here there is a more all India thing, because even though I have not visited parts of this, I'm definitely familiar with certain butterflies and certain moths of those regions. So I have helped in kind of detail. So these are the small things. And then if you are independently creating and publishing your small research data set, it could be even your master's project or even your BSc project. If you are collecting data in one place consistently, you can create your own data set and publish on GBIF. And then that you can even track exact number of citations and things. And your data set gets a DOI, you can put it in your CV with number of citations. So that is how GBIF is now helping and in some sense incentivizing researchers to publish more and more so that more and more people can use it for meaningful research. Yes, write back to me anytime, and we can not immediately, but maybe in November or December we can organize a little more advanced thing where we can learn how to do more advanced queries on GBIF data, how to download the data set, and how to start using it.